You know that feeling. You're standing in the kitchen, it's probably late, and there's a pizza box or, I don't know, a really nice cheese board in front of you.
Oh, I know it well, the late-night kitchen scenario.
And you tell yourself, okay, just one slice, or maybe just one little wedge of brie. You mean it too. You have every intention of stopping.
The intention is always pure, the execution, though.
Right, because 10 minutes later, half the pizza is gone, the cheese board is a wreck, and you're just sort of standing there wondering, what just happened? It's like a blackout.
It is a totally universal experience. And that's usually when the shame kicks in, right? We start blaming ourselves.
Exactly. We think, I have no willpower, or I just need more discipline. We make it a character flaw.
But what if it's not? I mean, what if that whole just one slice problem isn't about willpower at all?
What if it's actually biochemistry?
That is precisely the question we're digging into today. We're looking at a pretty dense stack of research, all centered around a report called Metabolic and Physiological Consequences of Beta-casomorphins.
Which is a mouthful. But basically, we were diving into why not all milk is created equal.
Right. We're talking about a specific genetic mutation in cows that might be turning some of your favorite foods into something that acts surprisingly like an opioid in your body.
The sources even have a name for it. They call it the dairy crack phenomena.
They do. And as we get into the science, you'll see that's not just, hyperbole. There's a real mechanism there.
So our mission today is to figure out if one tiny genetic switch in modern cattle is secretly sabotaging weight loss, messing with digestion, and maybe even your insulin levels.
To really get it, we have to zoom way in, down to the molecular level. We need to talk about a protein in milk called beta-casein.
Okay.
Bovine beta-casein is a chain of 209 amino acids.
Can we picture it like a charm bracelet, something with a bunch of links?
A charm bracelet is the perfect analogy. So you've got this long chain with 209 individual links. Every cow makes them.
Okay, 209 links. Got it.
Now, the entire difference, the thing that changes literally everything we're about to talk about, happens at just one single link. Position 67.
Position 67. So that's the smoking gun.
That's it. In the original ancestral version of milk, which we call A2, that spot is held by an amino acid called proline.
A2. So that's the old school stuff.
It's the original recipe. It's what you find in goats, sheep, humans, and older breeds of cattle like Jerseys or Guernseys.
Okay. But something changed.
Exactly. Thousands of years ago, a random mutation happened in Western herds, specifically the big black and white cows, the Holstein Friesians.
The ones you see everywhere.
Okay.
The ones producing most of the milk in the supermarket.
The very same. In those cows, which produce what we now call A1 milk, that proline at position 67 was swapped out for a different amino acid, histidine.
So we just swapped one bead on the bracelet, proline for histidine. Does one little change really matter that much?
It matters immensely.
Right.
And it all comes down to the bond. Proline creates a super tight, strong bond. Think of it like it's welded shut. It's very hard for digestive enzymes to break.
Okay, so it's a strong link in the chain.
But histidine creates a weak link. It's fragile.
Oh.
So when you drink A1 milk, your digestive enzymes, particularly one called pepsin, see that weak histidine bond and they're like, easy target.
They act like a pair of scissors.
They snip the chain right at that spot, and when that chain gets cut, something is released.
And that something is the problem.
That's the problem. The cut releases a tiny bioactive peptide fragment called beta-casomorphin 7, or BCM7 for short.
And A2 milk doesn't do this.
Barely at all. That proline bond is just too strong. But A1 milk can produce up to 4 times more of this BCM7 peptide.
Okay, so BCM7 is floating around our system after we drink A1 milk. Why is that bad?
Well, the clue is right there in the name, beta-casomorphin.
As in morphine.
Yes. It's technically classified as an exogenous opioid peptide.
An opioid. So we're talking about the same class of compound as, like, heroin or endorphins.
Structurally, it's very similar to the endorphins your own body produces for pain and reward. And just like them, it's hunting for one thing: opioid receptors.
And this is where the whole dairy crack idea from the literature comes from. It's not a metaphor, it's describing the actual biochemistry.
It is a literal description of the mechanism. Now, this BCM7 peptide is what's called proline-rich, which makes it chemically tough. It's hard to break down.
Okay.
If your gut lining is perfectly sealed, you might be fine. But if you have any level of intestinal permeability...
Most people call leaky gut.
Exactly. If that barrier is even slightly compromised, BCM7 slips right through into your bloodstream.
And from the blood, it's got a fast pass to the brain.
It can cross the blood-brain barrier, and once it's there, it binds to opioid receptors.
Okay, so what happens then?
Well, your brain has a natural braking system controlled by a neurotransmitter called GABA. GABA is what keeps your impulses in check. BCM7 hits those receptors, and it basically inhibits GABA.
It cuts the brakes.
It cuts the brakes, and when the brakes are off, you get a surge of dopamine, the pleasure chemical.
And that triggers the reward pathway.
It creates a powerful cycle of euphoria and reinforcement, which explains the pizza problem we talked about.
Because cheese is just super concentrated milk protein.
Precisely. To make cheese, you're concentrating the casein. So the dose of these casomorphins is about 10 times higher in cheese than it is in fluid milk.
Wow. So it's like a concentrated delivery system for this opioid peptide. My cheese craving isn't just me wanting a snack. It's my brain seeking a dopamine hit.
The sources even mention that when people stop eating dairy, they can experience withdrawal.
Actual withdrawal, like with physical symptoms.
Yes. Things like mood swings, irritability, depression, and of course, intense cravings. It's a classic addiction response.
Unbelievable. But the effect isn't just in the brain, right? The notes mentioned a traffic jam in the gut.
Yes, this is fascinating and maybe a bit troubling. Remember, opioids slow things down. It's why a drug like Imodium works to stop diarrhea.
Right, it slows the gut muscles.
BCM7 does the same thing. It binds to opioid receptors in your intestines, and it suppresses the release of something called acetylcholine.
Which is a chemical messenger that tells your gut to contract and move food along.
Correct. It drives peristalsis, so when you suppress it, everything slows way down.
How slow are we talking?
The research puts a number on it. A1 beta-casein can delay gastrointestinal transit time by approximately 6 hours compared to A2.
6 hours. So my food is just loitering. It's not digesting. It's just hanging out.
It's stagnating.
Yeah.
And you might think, okay, so what, I stay full longer? But from a weight management perspective, this is a disaster.
Okay, unpack that. Why is that a disaster? I thought feeling full was a good thing.
Well, if food sits in your gut for an extra 6 hours, that gives your digestive enzymes a much, much longer window to extract every last calorie from that food. It's a phenomenon called calorie harvesting.
So wait, you're saying I could eat the exact same meal as someone else, same calories on the plate. But if I had A1 milk with it, I might actually absorb more calories from that meal.
That's the theory. Your body is just becoming hyper efficient at pulling energy from food. And it gets worse.
Of course it does.
That stagnation, that traffic jam, it changes your gut microbiome.
How so?
It allows certain bacteria to multiply, and it can shift the balance toward a group of bacteria called firmicutes.
And that's the signature we see in.
It's the classic obese phenotype signature in the microbiome. These firmicutes are masters at harvesting energy from things you can't even digest, like fiber. So you get a double whammy, more time to extract calories, and bacteria that are better at doing it.
It's like your body's factory just went into overtime, but not in a good way. You're turning waste into waistline.
That's a perfect way to put it. And the sabotage doesn't stop there. It goes deeper into your cells. We have to talk about insulin.
This is where things get really heavy. The research talked about BCM7 messing with our ability to use sugar, and it mentioned molecular mimicry.
Right, it's a case of mistaken identity. The BCM7 peptide looks chemically very similar to one of your body's own glucose transporters, specifically one called GLUT2.
And GLUT2's job is?
It's the doorway that lets sugar into your liver and pancreas cells. But because BCM7 looks so similar, your immune system can get confused. It makes antibodies to attack BCM7, but those same antibodies can end up attacking your own GLUT2 doors.
Its body starts attacking itself. It sounds like an autoimmune reaction.
It is. And that's not all. It also interferes with a critical signaling pathway called PI3K.
Okay, PI3K. In plain English, what does that do?
Think of it as the delivery driver. Its job is to bring another transporter, GLUT4, to the surface of your muscle and fat cells. GLUT4 is the main door for sugar to get in and be used for energy. BCM7 blocks that PI3K pathway.
So the delivery driver never shows up. The door stays closed.
Exactly. Insulin is knocking, trying to get the sugar out of your blood, but the doors on your cells won't open.
So the sugar just stays in your blood.
Right. And your pancreas sees all that sugar and it panics. It pumps out more insulin to try and force the doors open. So now you have high blood sugar and high insulin.
Which is hyperinsulinemia.
Yes. And we all know insulin is the fat storage hormone. When insulin is high, your body is in lipogenesis fat storage mode. It physically blocks lipolysis fat-burning mode.
So even if you're eating at a calorie deficit, if your insulin is stuck in high gear because of this peptide, you're fighting a losing battle against fat storage.
Your body's own chemistry is screaming store fat.
That is just incredibly frustrating. And there was one more piece to this metabolic mess, right? Something about oxidative stress.
Yes, glutathione. The sources showed that A1 consumption leads to significantly lower levels of glutathione.
Which is the body's master antioxidant, the cleanup crew.
Absolutely essential. But to make it, your body needs an amino acid called cysteine, and BCM7 competes for the same cellular pathways that absorb cysteine.
So it's cutting off the supply chain for the cleanup crew.
Exactly. Lower glutathione means more oxidative stress. Your cells are essentially rusting. And when your mitochondria, your cellular power plants, are under that stress, they can't burn fat efficiently. Your whole metabolism just stalls.
Wow. It really is a perfect storm. Brain addiction, slow digestion, more calorie absorption, insulin resistance, stalled metabolism.
And then you combine it with gluten.
The pizza effect.
Yeah.
This was a huge aha moment in the notes for me. It says casomorphins from dairy and gliadorphins from gluten hunt together.
It's a powerful synergy because they're so similar. Gluten breaks down into a peptide called gliadorphin 7, which is also a proline-rich, opioid-like peptide.
So when you eat a slice of pizza, you're getting casomorphins from the cheese.
And gliadorphins from the crust. They both hit the same opioid receptors. They both slow down the gut. They both create that dopamine reward.
So it's not 1 + 1 = 2. It's an amplification effect.
It is. The metabolic damage is way more severe. It's why that combination of bread and cheese is so famously hard to stop eating. You're getting a double dose.
Okay, but let me play devil's advocate. We all know people who drink milk, eat pizza, and they seem totally fine. Lean, healthy, no issues. Not everyone is affected by this, right?
That's a crucial point. The key variable seems to be an enzyme called DPPIV.
And what does DPPIV do?
It's the only enzyme in our body that can effectively break down BCM7. It's your built-in defense mechanism. If you have really high, robust levels of DPPIV, you might be able to neutralize BCM7 before it ever enters your bloodstream.
So some people just have a better cleanup crew.
They do. But here's the catch. DPPIV is really sensitive. If you already have inflammation or a damaged gut, or even just due to genetics, your DPPIV levels can be low.
So the people who are already struggling, the ones with inflammation or gut issues, are the ones who get hit the hardest.
It creates a vicious cycle. Inflammation lowers DPPIV, which lets more BCM7 in. The BCM7 then causes more inflammation, which lowers DPPIV even further.
That makes so much sense. And there was that interesting note about the calves, too.
Oh, yes. The animal studies, they found that calves fed A1 milk grew faster and fatter than calves fed A2.
Which is a huge economic advantage if you're a farmer selling cattle by weight.
Precisely. But if you're a human trying not to gain weight, drinking a formula literally designed to rapidly fatten a baby cow, you can see the conflict.
So let's bring this all home. We've gone through a lot of biochemistry. We've got this BCM7 peptide acting like an opioid, slowing our gut, spiking our insulin. What's the practical takeaway?
The main takeaway from the sources is that this isn't an indictment of all dairy. It's a specific issue with the A1 genetic variant.
So the A2 milk, the ancestral version, doesn't have this problem.
Correct. Because of that super strong proline bond, it doesn't release BCM7. So the experiment for someone listening who thinks this sounds like me is to try switching.
Look for milk that's actually labeled A2.
Or look at goat or sheep dairy, which are naturally A2, or, plant-based milks to just sidestep the whole casein issue.
And this isn't just theory anymore. They're actually testing it, right? The MPACT study.
Yes, which is really exciting. It's a proper randomized controlled trial using DEXA scans, the gold standard for body composition, to directly compare A1, A2, and oat drinks. We're finally going to get definitive human data on this.
I cannot wait for those results. But for now, it really does seem like there's enough evidence to warrant a personal experiment.
Absolutely. If you're struggling with stubborn weight, digestive issues, or that feeling of brain fog after you eat, eliminating A1 beta casein seems like a very logical thing to try.
It just reframes the whole conversation. It shifts it from I have no willpower to
I am fighting a powerful biochemical signal.
and that takes all the morality and shame out of it.
It does. It makes it about chemistry, not character.
I guess I want to leave everyone with a final thought to chew on. We spend so much energy beating ourselves up about food. But if certain foods are chemically engineered by nature or by accident to bypass our satiety signals and hijack our brain's reward center, just like a drug, does willpower even stand a chance? Or is it maybe time to just change the chemistry of what we're eating?
That's the question.
Thanks for diving deep with us today. We'll catch you on the next one.